Chris Madsen is currently an associate professor in the Department of Laboratory Medicine at the Lund University in Sweden. And for all of us that didn't know that, a funny fact, Chris now lives in Copenhagen and just takes a bridge to go to Sweden. Chris did two postdocs. One was in the laboratory of Erik Sahai in UK, and then the second one is in Janine Erler lab in Denmark. And he opened his own group in 2016, still maintaining the collaboration with Erler in Denmark, where he lives. And the group of Chris Madsen is aiming to understand extracellular matrix remodeling during tumor progression at the single cell resolution. And all this is in the aim of stopping the metastatic spread by targeting either cancer cells or the tumor microenvironment. And today he's going to tell us something about his efforts on decellularization of the tumors and looking into 3D structure of extracellular matrix with 2-photon microscopy. So Chris, thanks for coming, and take it away.
Thank you so much for the invitation. I'm very delighted to be here. So today I'm going to tell you a bit about a particular project that I'm doing in collaboration with the University of Copenhagen, in particular with Alejandro Mayorga-Gugliani here, which I've mentioned because he has been as detrimental for this project as myself. So he deserves as much credit for this. The difference is that I've been standing for the imaging, and he's the veterinarian or the doctor, which are taking care of animals and surgery. So this is a full collaboration with the University of Copenhagen. I'm going to talk to you today. And it's about trying to understand how the ECM actually looks like in 3D in disease and in particular in cancer. So just to start with the definition is that the extracellular matrix is a collection of extracellular molecules secreted by cells, and these provide structural and biochemical support to surrounding cells. So this is the scaffold of any organ, any tissue we have. Without the matrix, without the extracellular matrix, we don't actually have a body, we don't have any functional organs. So the normal architecture of ECM and tissue is actually believed to act like a barrier for tumorigenesis. That means actually a healthy tissue is actually preventing transformation and neoplastic growth. While at a certain point during the disease progression, the environment gives up and a lot of changes happens during dysplasia, and the tissue starts actually promoting the cancer. This is the way we believe it's working, and this was actually already proposed more than 10 years ago by Nina Wiesel and company and collaborators. So the aberrant ECM, as we know, we believe in most cases are contribution to cancer pathogenesis. Although in certain cancers like pancreatic cancer, there are strong beliefs that actually the matrix may actually be tumor restraining. And I'm not going to go into detail about this, but just to let you understand that the matrix has many functions and can potentially be used in both treatment, but also in diagnosis in diseases and particularly in cancer. So the ECM has prognostic value, it has this gene signature, and it can be used to predict cancer outcome. However, how does the actual ECM look like? And this is a classical image that you will be seeing in 99.9% of papers. And you know, it's not really high resolution. It doesn't really give you a nice structure. And if you're lucky, you get an image like this, which looks good. You know, you have some different capillaries, you potentially have a vein going through the tissue. But again, you know, majority of this tissue goes down to, as being a section to between 4 and 10 microns thickness. And if you know how big a vein or an artery is in a mouse or even a human body, you'll quickly realize that 4 to 10 microns is not enough to even cover one single vessel. So meaning that we will lose information on every single aspect of this 3D structure. So what happened was that when I joined the Janine Erler's lab, I met this crazy Venezuelan doctor, Alejandro Mayorga-Gugliani. And he had this idea that one day he came with this to me. And he said, look at this, Chris, he said to me, what is this? And this was a mammary fat pad. And then he showed me this, and this was obviously a lung. And then he showed me this, and I was a little puzzled that there was obviously a couple of sentinel lymph node on a string. And then he showed me this, it was a melanoma cell growing inside the tongue of a little mouse. And he told me, what is this? And he said, I removed all the cells from the tissue, leaving just the extracellular matrix scaffold. What should we do with this, he said. So this crazy doctor has been operating all these animals, has been able to remove every single cell in a tissue and potentially remain and maintaining the structure of the organ. So as he told me, Chris, what should we do with this? And I was always into imaging, and I was always into extracellular matrix and tumor migration. We decided to collaborate with Thomas Cox here, and as the three handsome men here, the bad, the good, and the ugly, you can decide which ones are what, got the opportunity to work together and try to get this project ongoing. And Janine Erler at the time, our boss, she said to us, just do whatever you want, I trust you. And we initiated this collaboration some years ago. Tom was doing the proteomics, I was doing all the imaging, and Alejandro was doing animal work and decellularization operation. And together we were conducting this project. So this data has been published a few years ago, and the technology we developed, we called it ISTOT, for in situ decellularization of tissue. And pretty much what happened is that you take any organ of your likings and you actually decellularize it by removing all cells, and you have empty organs full of extracellular matrix proteins that you can use for proteomic analysis, multiphoton microscopy, and even later on as a bioreactor where you repopulate these organs. So the technology we used here was actually to do advanced operation on these organs, thereby using a very low detergent to flow with a peristaltic pump to flow it through the organ of interest. And in this case, it could be mammary fat pad. It could also be a lung, it could be the brain, it could be the liver. So in each case, you have to direct the flow to the specific organ with a light detergent to kill the cells. So we managed to decellularize up to 33 different murine tissues, and this was published in 2019, and this is just to give an example. And in many of these cases, the operation you need to do on the animal is different, as we have to target the different organs of the tissue. And as you can see here, we have the ovary, we have the rib cage, we have lung, of course, we have the eye, as you can see here, and so forth and so on. So the first thing we needed to do, obviously having taken all the cells out of an organ, was of course to characterize this tissue. And first of all, we compared it using second harmonic generation imaging. As you can see here, we have the mammary fat pad. This is a fresh one. And underneath here, you have an ISTOT, which is the decellularized organ. And we compared mammary fat pad, lymph node, and lungs, and later on many organs. And we could quantify that there was actually no differences in the orientation of the fibrillar structures we saw in the tissue. And we could also do a gap analysis to see if our tissue was either expanding or collapsing while we were doing this. And in all cases, it looks like the structure was maintained. And we could also determine that the fibrillar diameter in the different organs was maintained at approximately 1.5 microns in the animal. So the next question we will ask us, what about the vessel integrity? Now we have removed all the tissue, what happens to the vessels? Are they collapsing? What's going on? So for that case, we actually did a trick. We used a heated corrosion cast, which we injected into the lung tissue, for example, here. And as you can see, that the lung capillaries and the vascular network was maintained, and we could see this plastic going through all the capillary, all the blood veins, blood arteries in the blood, ending up in the smallest capillary at the end. And this is an introduction. This is where we have actually removed the lung tissue and just have the plastic cast left. When we injected dextrans, we could actually also see that the dextran was maintained in the tissue. Actually very surprising, because you will actually imagine when you remove endothelial cells that the dextran will leak out of the vasculature very quickly, but this was actually not the case. So it's really suggesting that the basement membrane is actually a very important factor in leakiness of vasculature. The next thing we did was we tried to take different kind of organs, and here we took a lactating mammary gland, and as you can see, this is the entire gland we have here, and we stain this for collagen 4, and we look at the collagen fibers, and we could quantify here, as you can see during lactation, is that the collagen fibers start to get linearized compared to the more curled and bended collagen fibers we see in a healthy or normal, let's say, breast. We could also look at terminal peripheral nerves. So this is a nerve which runs through the tissue, and this is a maximum projection of 3D scan. But if you look into details, you can look how the nerve end is running with some vasculature around. And if you go into details inside the nerve, you can actually see how individual nerves are covered, and you can see where the Ranvier nodes is, where the myelin sheets are closing around the nerve things here. This is an image of the kidney structure, where we can see all the different basement membrane of the kidney structures. You see the vasculatures, you see the arterioles, the glomerulus, and so forth, and the Bowman capsules, and all these things could be imaged very, very high resolution. But when I started to go a little bit in 3D resolution and start to understand how these different vessels looks like, it became apparent that vessels are very different. We thought a vessel is a vessel, but it's actually not. So this is to show you here a little artery. And as you can see, this is a very beautiful artery, which is covered by a second layer of basement membrane, most likely produced by the pericytes of the smooth muscle cells, with a very, very specific distance of 5.9 microns all along the artery. But the vessel next to that one look very different, clearly showing that vessels are differently organized with respect to basement membrane proteins. And this was even more evident here when we looked at the vasculature in the eye. And here you can clearly see that the collagen force and the laminin expression in the different vessels are looking very different. And this was surprising us because it's always been the conception that all basement membrane has the same level of collagen 4 and laminin and it's all identical, but this is clearly not the case. And we can clearly see here that arteries and veins and capillaries have different expression level, or at least different epitopes recognized by these antibodies in the tissue. We then went into a bit, so I don't know if you can all see these kind of movies because this might be a little bit heavy to some people, but when you go into 3D reconstruction of these kind of vessels, we can get really nice images of how vessels and structures and different ECM components are actually organized inside the tissue. So we have now also been able to validate more than 35 different ECM antibodies, and we published this in Nature Protocol, and these are the first catalog of ECM proteins which are really being validated to work in mouse. Most of these antibodies have been validated also in knockout mice, so it's a really good catalog, and I really suggest people to have a look at this list to see if a specific antibody or specific protein of interest is among this list. We'll be happy, of course, to validate other antibodies as well. But it is clearly that these antibodies detect different organs, different structures in the tissue, as shown here in some different kind of organs. Now we're trying to, of course, go a little bit more in more detail. This is a huge 2.5 millimeter section, a tile scan of the heart of a mouse, and this is just one section through, and you have different kind of let's say organ and tissue in these things, and it's of course very disorganized. But what we're trying to do is to build big maps in high resolution using multiple tiles in that. But before going to these kind of things, I would like to emphasize a little about how do we know and what happens in cancer. So we start looking a bit at the primary tumor. As I showed you before, we know that these structures in the fresh organ and the isthmus, the mammary fat pad is beautiful, bended or curly collagen structure which become linearized as the tumor grows. This was also shown by some of the images before, and we can quantify this very nicely. And this is another imaging of a little mammary tumor growing in the mammary fat pad, and you can clearly see that around the mammary fat pad and inside the mammary fat pad we have linearized collagen, while out here in the border and the edge we have these curled structures. And this is interesting because you may have asked yourself, what's the difference between these curled structures and these linearized structures? And this was actually postulated to be published last week, a very interesting study by Claire Waterman, showing that that curled structures actually seems to, and benign ECM seems to be inhibiting tumor cell migration simply by, let's say, screwing up the polarization of the atomizing and contractile networks in the cells. So when the cells are moved along linearized collagen or as in a 2D surface, you have a polymerization of the cells which nicely migrate and polarize actin filaments in the leading edge, while a cells moving and touching along curled bended structures actually suddenly seems like polymerized actin in different direction, thereby depolymerizing the migration and the directionality of the migration. So they proposed in a very nice things that curled and benign ECM architecture inhibits tumor cell exit from the tumor, while the linearized obviously promotes directional migration and tumor exit through regulating of focal adhesion, integrin signaling, and ectomizing contraction. So when we went back to the tumor, saw is that we actually not only did see things in the tumor, but also adjacent to the tumor. And this is some vasculatures in the mammary fat pattern. What we very surprisingly saw is that the collagen structures here starts to get a little bit disorganized. They're not completely linearized. It's adjacent to it. But you see this kind of strange resolution and secretion of basement membrane protein in the tissue. And if you look at this little area here in this box, we saw some very surprising changes. We saw complete resolution and loss of some basement membrane. We could even see holes in this tissue. And when we measured these holes in these vessels, it was exactly the size of the cells moving through this tissue, suggesting that potentially immune cells, other cells, tumor cells moving through the basement membrane actually leaves holes behind them, which obviously need to be repaired later on. The basement membrane fibers we, for the first time, actually observed outside the basement membrane. So this is a collagen 4, which is tend to be believed is to always be associated with the vasculature or the epithelial layer. But here we saw tons of small filamentous fibers all over the tissue, which we have never observed. And in the beginning, when we showed this to ECM experts, they didn't believe us. But when we showed them with the 5 different stainings using nitrogen, collagen, laminin, they start thinking, well, maybe this is true. And this is a very interesting observation because this is something we only see on a very dramatic inflammation on a cancer, potentially suggesting that some cells could follow these tracks into the tissue. When we took an entire sentinel lymph node and we scanned through it, we can kind of see here that this is the healthy lymph node, and here's the disease one. We have a micrometastasis growing here in the lymph node, and we can see these kind of structures. And when we go a little bit in further detail and do 3D reconstruction, we can get this kind of maps in the lymph node. The beautiful thing about this technology is that we can image the entire lymph node from one end to the other in a mouse. But obviously, this doesn't really give us high resolution. So what happens if we zoom in on one of these tissues more here, and we start looking at the vasculatures inside the lymph node? And as you can see on the left-hand side, we have some nice, beautiful vessels here. They look very smooth and nice, and we have some capillaries running through. And when we have a cancer metastasis in the lymph node, we see this dramatic remodeling. First of all, as I showed you, these small capillaries all over in the tissue. But as you can see here, the vessel starts to be on fire, and you have this sprouting phenotype of cells actually potentially laying down basement membrane proteins outside the vessel. And this is to open up. And you could even zoom even more here, and you can really see that the basement membrane, the vessels really have cracks in it. They're really leaky, and they are not healthy at all. So when we looked at these things and the differences, what happens into the metastatic lung was one of the first questions we asked ourselves. And here we looked at the very small macrometastasis. So we're talking about having, like a 200 microns in diameter. We're talking about maybe 50 cells. And the first thing we could see was immediately just by looking at the second harmonic generation here is that you have a little crater there where a tumor has begun to start to grow. And this obviously also affected the elastin fibers that you get automatically when you image these tissue. Clearly suggesting that small macrometastases are remodeling and potentially pushing away the collagen fibers and the elastin fibers in the lung tissue. However, when the tumors start to grow, as you can see here, this is a fresh tumor, and this is kind of, let's say, the macro tumor, macrometastasis compared to the healthy tissue. We could see a massive difference in the structure of the collagen. So again, here we have a beautiful collagen structure in the cortex, and when we look into the metastasis, we have this linearization of the collagen, which is dramatically altered in these tissue. So these kind of experiments suggest that the macrometastasis and the micro and the macrometastasis develops and changes the collagen structures as shown here, that you begin having a small crater, a small remodeling, and as time goes on, the tissue really changes over time. Interesting, we have good evidence that cancer-associated fibroblasts may be responsible for this linearization of the collagen fibers, but this is something for another story. When we looked into this macrometastasis, as you can see on to the left, this is inside the metastasis, this is the healthy tissue, and we start looking at the capillary networks in the alveoli, and we start zooming in. You could clearly see that the beautiful alveoli structures are looking like this, and as you go through the invasive front and the front here of the macrometastasis, disorganization of the vasculature inside the tumor occurred, while you also seem to have like a secretion of additional basement membrane protein disorganized here in the invasive front, clearly showing that the blood vasculature is altered in these macrometastases of the lung. So of course, what we also could do, and this was performed by Tom Cox, is that we can of course run let's say mass spec proteomic analysis of these ECM proteins. I'm not going into detail, but what we could detect is of course massive differences in structures of ECM proteins in different organs. This is just to give you an idea. For example, if you take von Willebrand factor, you can see this is down in the metastatic lung, but it's up in the lymph node and it's down in the tumor, versus tenascin C is up in the metastatic lung, it's up in the primary tumor, but there's no difference, or even down in the lymph node. So each actually ECM protein has a different kind of prognosis or maybe even diagnosis which can be used. And the question is whether we can use these kind of things in the future as a theranostic. For example, could we actually treat, for example, a tumor here with the collagen 12A because it's eightfold up compared to, for example, metastasis. So what we did some years ago is that Alejandro began to design, we wanted to actually start to repopulate these tissues, and we made a bioreactor. And then now Alejandro has a talented PhD student, Maria, which is working on this project, and she's developing much further than I'm just going to show here. But the idea is that you start decellularizing an organ, as you see here, is completely decellularized after 24 hours, and then you can actually pump back cells into this organ. And what I'm going to show you here is just a couple of movies where we injected tumor cells, and after 1 day they follow the capillaries. But as a couple of days after, these cells start to migrate, they start to move around, they're happy in the tissue, and we can image these things in this bioreactor for many, many days and look how these cells happens. And just to give you an example of high resolution, here we have some division going on in the cells. Here there's a cell division going on there. Cells are moving around. So this is the nucleus, which is marked, and we get another division coming up in a second over here. The cells are happy, they're moving around. What we also can see is, for example, nuclear deformation. You see one down here in the corner. We will see another one, nuclear deformation. So even though we only see the collagen fibers here, we can obviously know that many other ECM components are there. But Maria, she's working on this concept, and she's having a paper in submission. I'm not going to talk about it, but she has been repopulating these organs with T cells, cancer cells, fibroblasts, and so forth, and been doing a lot of cool stuff that hopefully within some months will be published. So the big question came a few years ago, can we actually decellularize human tissue? And this is really what we wanted to do. You know, we had the proof of concept, and we began by simply getting some biopsies from human breast. And this is like a huge human breast. This is a one time centimeter biopsy from a human, and this is a tile image of 433 images. And you can really see how interesting the collagen structures is in these kind of tissue. And we start zooming a little bit in here, you really see how the collagen fibers in this human patient is altered. And as we zoom in, you can really see how the fibers beautifully align in the tissue. And if you compare it with the mouse collagen fibers and mammary fat pad, that's actually a certain amount of similarity. Probably the biggest difference in the human tissue is that we seem to have many more fibers kind of like cross-linking next to each other in a curled bundle form compared to the murine collagen, suggesting that probably the diameter of, let's say, bigger collagen bundles are much broader in humans. We then started going into the lung imaging, and this is one of the first images we did. And here actually what you can see, which is the fantastic thing of lung, in human lung, is that the elastin fibers is dramatically fluorescent. So compared to the murine lungs and the murine tissue where we have a faint autofluorescence, human elastin is so bright that you barely can do anything else than looking at these things in the tissue. And this is what you see, this huge bundle of color of elastin. So this is an image of one alveoli in a human being, right? So we went into this kind of high resolution imaging of human tissue. And this is a patient with pneumonia, and we could start organizing, looking at different parameters like the second harmonic, so fibrillar collagen, elastin fibers, periosteum, collagen 4. And these studies has now turned out to be an extremely powerful things, and Alejandro is working very hard with pathologists of the hospital to try to organize these structures in various different human patients with different lung diseases. And this is to give you a little bit of just, this is a small movie. I don't know if it stopped already there. I didn't get it, so I will not go through. But this is, of course, done in 3D reconstruction, and we can now go in and watch these things. I think my computer also froze here. I'll give you a second. This movie was too big. Here, oh, sorry. We have to go one back. Now I have some movies that are too big for you. Give me a second. So I can finish off here, but I can just show you that this image is, for example, an image of some of the patient that Alejandro and collaborators has been looking at, and this is to look at the human parenchyma. And here you see the beautiful alveoli structures. This is the tile images of 1400 images, and you can see as different human diseases how the structure not only of the elastin and the collagen fibers are altered, but you also see other alterations like periosteum and so forth. The biggest difference here is that in some diseases we have what we call a tremendous fibrotic tissue of second harmonic signals of fibrillar collagen, and in other diseases we seem to have a very, very strong changes in elastin structures. And of course, the big thing is, of course, to try to go in 3D reconstruction and make movies of this. So this is inside the human lung, and these kind of dots you see here, this is the tar from a smoker, has been smoking too many French cigarettes, maybe, I don't know. And you can really see how the elastin fibers, how the different collagen fibers are really moving into this tissue. So this movie I took in a few years ago, and I think this is going to be the future in our science, to get this kind of resolution and this kind of imaging. So this is a 500 times 500 times 500 micron cube of a human lung, and we are now trying to minimize this a bit because this took 3 hours to take this image, and we're trying to see if we can implement this kind of imaging to lung tissue and try to figure out if we can use this for diagnosis on patients. So in summary, we published these 2 papers. You're very much welcome to contact any of us if you're interested in the technology. But in summary, I want to tell you that this ISTA digitalization actually seems to preserve the ECM and the basement membrane integrity of any organ we have studied so far, even in human beings. I don't know why this jump one, but what we can do is that the big difference here compared to tissue with cells is that we have great antibody penetration. So we don't have any block of the antibodies. We can penetrate any tissue, and we can get the antibody in and bind all this its epitope. So we have great imaging penetration as well. We can image much deeper than if we had cells, and we can thereby reconstruct 3D topology of huge amount of tissue. As I showed you before, the video was high resolution in a 500 micron stack. And we can, of course, quantify biochemical composition and so forth. We can use this as a bioreactor. So Marie and Alejandro is working hard on granularity in this bioreactor and trying to do various different experiments in that. Human tissue can be characterized with ultra high resolution, and hopefully maybe in one day ECM patterning could be developed into new prognostic, diagnostic, and theranostic tools. Partly at the end, Alejandro and people in Janine's lab are working very hard on trying to incorporate these things into machine learning and artificial intelligence to understand how the extracellular matrix and the structure in the human tissue, in particular now in the first paper here, soon to be submitted, how this is going to be useful in pathology. To determine which kind of, for example, fibrosis or COVID-19 or pneumonia you have in the tissue. So with that, I think I will acknowledge the people which I did all this work with. So all of this work was pretty much done in Copenhagen. I did some of the last part of imaging, 3D imaging was done in Lund. Alejandro Mallorca, detrimental for this project. He developed the digitalization method. I did all the imaging, but of course, without the support from Janine and all the members of her lab, and all the funding that Janine just supplied us with, we will never been able to perform this kind of work. I need to thank the Center of Imaging, but I also need to thank Leica for giving me specific access to some of their high-resolution microscope in Mannheim in Stockholm. So with that, I will take any question, and thank you for your attention.
That was a fantastic talk. Thank you so much.
You're welcome. Should maybe, I stop sharing, maybe?
Yeah. I have a question.
Yeah.
Buena, yes.
Yeah.
But, so when you are decellularizing the healthy tissue, I think that was the mammary gland, I think, and putting back, I mean, you have the squiggly, disorganized extracellular matrix. If you put back, you recellularize with either wild type cells, tumor cells, or fibroblasts, which cell type would be responsible for remodeling of DCM? Do you see actually the remodeling in this case? Do you see this linearization of collagen fibers, and which cell is doing this?
Yeah, so Maria Rafana has a project ongoing when they try to understand by injecting normal fibroblasts and CAFs and T cells and so forth. And what we believe is ongoing here is that fibroblasts are detrimental for this remodeling. And we see some differences when fibroblasts are present with respect to not only ECM, potential ECM degradation, but also the linearization of the collagen. But I have to say, I think much more work is needed for that. I mean, we know macrophages are important to stimulate and activate the fibroblast to then secrete and remodel. So I think this is a process in concert with different cell types. And there's also a very important aspect of this is when you do this kind of bioreactors, time potentially will also, like expansion of the tissue potentially. What we don't know is how much does that actually alter the structure when we are imaging for example, 2-3 weeks, right? This is another thing, pumping constantly media and growth factors and hormones to the tissue. So I think it's probably not the best model to use for determining which cell type, but it's clear you can make it more simple by injecting one cell type at a time, but I don't have a definite answer for that. But fibroblasts are clearly important.
Thank you.
Yeah, so I'm wondering how different is it still has to be different extracellular matrix when the tumor cells and all the other cells of the microenvironment are present in vivo, because tension is there. There's a lot of pressure that's exerted by the cells that are present. So there has to be some relaxation going on once you decellularize. So is it even technically possible to image in vivo or ex vivo at least, and then re-image the same tissue post decellularization and compare the structure?
So that when we compare the structure of a fresh tissue and the structure of the decelled tissue,
right? But the same exact regions, I mean, or I guess you do the whole.
Yeah, that could be interesting to do. But again, I mean, we've done many quantifications, right? And we optimized many different detergents, and some detergent clearly changes. Like you should never add SDS on Triton-X and something like that, because you get an expansion and degradation of the collagen, and the diameter increases and so forth. But if you look at the gap analysis we did in the lung, which was very surprising, is that the gap analysis is nearly identical, right? So we don't seem to have a huge expansion or a huge collapse. It's clear that the cells generate tension, like compression in the tissue, and it's also clear that the tissue is softer. So if you did a bulk atomic force microscopy, or let's say shear rheology of the entire tissue, it would be softer. But the question is whether the individual fibers are using atomic force microscopy will be different from the two different cases. And I'm not convinced that the individual fiber is going to be much different. but the bulk stiffness, the bulk tension will of course be very different. This is clear. And when we do proteomic analysis on a D-celled organ and a normal fresh organ, we see the same protein, practically speaking. There's huge overlap. What we do lose a bit is some of the growth factors and cytokines, which are inherently difficult to actually detect by mass. Right? So that's even, you know, we can't even know how much we're. And we have not looked properly at sugar molecules. We don't know how they look in the tissue. That's another thing.
So I guess by fixing, so you're fixing by perfusion, right?
Not fixing anything.
You're not fixing at all.
No. There's no fixation here. It's totally. That's why we can put cells back in and they can move around and remodel and migrate and dig around, you know.
I'm still struggling to understand, like, how is it possible that the tissue doesn't relax at all? So because, you know, the work of Rakesh Jain on like the tension coefficients, like that somehow is inconsistent with this. So,
but you know, we use flow constantly, right? So we have, you know, let's say flow to maintain. We constantly have a flow on, right? So when we have a flow in, it's like, you know, perfusing an organ before fixing it, right? You have a flow in, you have a flow when you have cells in the bioreactor. We keep going. We have an inset and an outlet and so forth, and this is how it works, right? So
I see. So this is how you maintain the tension, actually.
To some extent, right? But of course, it's not the same kind of as having cells in there. But
right.
That's why its vasculature is not completely collapsing. And when we inject cells, the first day the cells are only sitting inside the into the, let's say, empty vessels. And after a day, they start to migrate out of the basement membrane and into the parenchymal tissue. You know, this is what happens.
Yeah. So practically, your work is saying that most of the extracellular remodeling is permanent and based on the molecular changes, and not so much on the physical and mechanical.
Oh no! I think that I will not draw that conclusion. But because of course the mechanical changes are there, you know, the blood flow and so forth. But we do maintain the blood flow, right, to some extent, not the same. But the biochemical properties are, of course, the ones which maintain the biophysical properties in the tissue, I would say. It is, of course, not 100% the same as in the tissue, but this is not the point of this technology. The technology of the point of this technology is to produce high 3D resolution imaging of the tissue. No one has been able to image proper vessels like in 3D reconstruction, right? And now we have a chance, and just by looking at a big area, you can see that all the vessels looks different, right? Depending on whether you're an arteriole, an artery, a vein, whatever you are. And these are surprises that no one actually knew, I think, before we start doing this imaging.
Okay, and sorry, Daniela, you have.
Can we jump in? No, it's one question from the Q&A, and then maybe we should move on, Buena, because it's quite late. So you have a question from Eric Sahai. He's saying, very nice talk, Chris. How do you perfuse detergent in the patient samples? Which is probably related to Buena's questions.
So this is the same. So of course here you don't have, you can't use the natural physiology from the patient, or you don't have a heart or lung. So what you do is that you find vessels that you can perfuse into the organ. So if you get a biopsy from the lung or from the brain, whatever, we have to operate so we can find vessels, and we have to pump it through and you get exit. And sometimes we have to clamp some other vessels so you maintain a proper flow into the tissue. And I have to say, this is not something I'm able to do. This is all Alejandro's work. You know, he's the microsurgeon. And I mean, if somebody wants a microsurgeon to do stuff, you should get this guy. It's amazing. So he's looking for a PI position. So everyone contact him, right? He needs a PI position. So he do all microsurgery. He knows all the vasculature of an animal. He clamps whatever he needs, and now he does in human as well.
Okay, we have one more question from the panelists. So Kenneth, our next speaker, is saying, during the repopulation experiments, do you observe cell migration polarized along the collagen fibers, or are cells agnostic to fiber direction?
And so this is a good question, because of course, what you just saw was the second hormones of the fibrillar collagen, right? But there's many more fibers in that tissue. There's connection fibers, there's connecting fibers, you know, and tenacious, whatever. But what we do see actually sometimes if you shoot the movies is that you can actually see that they're migrating along some tracks, some collagen fibers. But we can also clearly see by this movie that they are not only migrating along these tracks. That was clear to see. So collagen fibers are clearly there. They are sometimes coated with other proteins, but we do also see migration along other paths than the fibrillar collagen.
Okay, thank you so much, Chris. Kenneth received his PhD in Stanford in biophysics in the lab of Manish Bhutt, and at the time he was developing new technologies that combine atomic force microscopy with fluorescent microscopy. And right now he's at UCSF in Max Krammel's lab as a postdoc, where he's developing new technologies to dissect the role of tumor infiltrating immune cells and how they speak their crosstalk with the spatial heterogeneity in immune cell composition and expression state. Okay, I think we're going to hear the details of that coming up. So, Kenneth, you can share this whenever.
Yeah, thank you for the introduction. And thanks to everyone for being flexible on the time for us Americans. Yeah, so let me begin. All right, and thank you also to the organizers for giving me this opportunity to talk about the work we've been doing in the lab. Basically, our goal is to be able to align the kind of very high dimensional data you get from single-cell RNA-Seq and other potential single-cell omics technologies, and combine that with the kind of three-dimensional space information you get from imaging of tissue sections. So today I'll be talking about how we've applied this to tumor models in order to understand the role of the very heterogeneous population that is the tumor-associated macrophage, and understanding where they sit in the tumor and potentially what roles they play in the tumor progression and the immune response to the tumor. So to start with, I think we can all appreciate from imaging the tumor, especially ones that spontaneously arise, that a lot of tumor heterogeneity kind of involves. And there's a lot of spatial heterogeneity, of course, in the tumor, with variables such as, well, the local ECM architecture, as we saw before in the previous talk, with those very nice images, and the degree of infiltration of certain immune cell populations, and kind of which cells are closer to which other cell types. So ultimately, we'd like to understand, how a cell's expression profile is actually influenced by its position in the tissue, its XYZ position. So this example of like two-photon imaging of human CRC biopsy, and you can see there's areas of increased collagen buildup, and there's areas where there's increased clustering of certain cell types, in this case CD8 T cells, and increased clustering of myeloid cell types as well. And of course, I'm sure everyone is also familiar with the explosion of single-cell technologies, including single-cell RNA-Seq, and you can generate, this cells in transcriptional space, but then oftentimes you don't really know where that cell came from in the tissue. So being able to kind of map the cells, these dots that you see in the UMAP, back to an actual position somewhere in this imaging setup would greatly enhance our understanding of the kind of evolution of these of spatial heterogeneity in the tumor, and thus allow us to kind of link expression space with physical space. So this approach, which we term ZipSeq, it's published in Nature Methods, so you can read up on the details. So I'm not going to spend too much time on the actual mechanics of it, but the general gist of it is to basically create a regional hashtag in an intact piece of tissue. intact tissue section. So when you image your tissue, you may see regions of interest, areas with some increased cell clustering of a certain cell type or aberrant structure, or et cetera, et cetera. So this begins with basically tagging your cells of interest. So in this case, everything you see is done with an anti-CD45 antibody, allowing us to basically tag all immune cells. Of course, the anchoring moiety can theoretically be swapped out. So once you've stained all your cells, once you've labeled your cells in your section with this antibody oligo, you're able to basically define a region of interest, illuminate it, and then basically these photo cages fall off. And with the addition of a readout oligo strand or a zip code strand, so in this case, zip code 1 will hybridize, then we can wash that away and repeat this n times for n regions theoretically. So now we have cells in one region barcode with this orange barcode, as you can see here, cells in another region barcode with this purple barcode is in here. And of course, upon tissue dissociation and extraction of single cells, we're able to basically generate kind of two parallel libraries. Anyone familiar with like HashSeq or something will immediately understand what we're doing here. You basically have a cDNA library for every cell and potentially a zip code library for every cell. And this allows you to map back that transcriptome, that single cell transcriptome, to that region you just defined in that section. All of these sections can be done, and you can thus acquire multiple regions of interest on the same section at once, and then basically start to overlay that spatial information on top of the transcriptional information that you normally receive. So this was just we just covered in the paper. So again, I'm going to go through this part relatively quickly. In this, we actually use an orthotopic PYMT breast cancer model, this CHOVA model. And then we also transferred in these tumor-specific CD8 T cells that are GFP positive. And then we can harvest the tumor and section this. And what we see by imaging, unfortunately, our images won't be quite as pretty as the one before, as the talk before, but as you can see, you have your tumor mass, and then kind of trapped on the rim are these clusters of those tumor-specific GFP T cells. And then there are also T cells that further infiltrated deeper into the tumor space. We can define, in this case, two regions, kind of what we define a rim or outer and inner, and then you can overlay upon single cell dissociation and then your classic encapsulation, you can actually see that there are different your major immune cell populations, but you can also see that they have different regional compositions as well. And what we saw in this particular instance was this kind of increase in, preferential localization, monocytes to the exterior, and a preferential localization of macrophage populations towards interior. And we also saw was this kind of shift. If you subset on those GFP positive T cells, you saw the shift in basically a more stem-like to more exhausted signature based on kind of depth of infiltration. And this is kind of, you can see this nicely in these violin plots here. And also the overlay of these scores. And then of course you can perform differential gene expression between cells that are denoted margin or interior, and find a lot of these classic genes associated here with more terminal exhaustion, here more stem-like state. So this was our initial pilot study done with two regions. What we wanted to do was basically extend this to further define more regions at once and use it in a variety of different models. So from here, we were quite interested in this macrophage story. So then this brings up the question of what a tumor-associated macrophage is. And as you can see in this nice graphic, there's this kind of canonical M1 to M2 idea. However, I think the field is moving away from that now, realizing that rather than just lying along a continuum, this bipolar continuum, they can occupy a very diverse set of states. And what we wanted to understand was, first of all, what states exist within the tumor, and then second of all, within a spatially heterogeneous tumor, where do different TAM populations reside. And if we can understand that, we can start to look at their neighborhood, what other immune cell types live in that neighborhood, and thus try to figure out how the local environment may shape a TAM fate, and vice versa, how TAM state may shape their local environment, that is, the cells neighboring them. So if, right, and then in the future, we plan to extend this analysis to CD45 negative cells or non-immune cells. So in order to get visual regions of interest that are kind of necessary for this approach, we actually built a reporter mouse for CD206 or MRC1. That's a common marker used to denote this kind of M2-like state. So this reporter was built by basically inserting this LSL reporter cassette into the CD206 or Merck1 locus, and then crossed with the CSF1R Cree. This actually results in this CD206-driven expression of Venus and also DTR for potential depletion experiments in CSF1R expressing cells, so theoretically your macrophage lineage cells. And then if we perform 2-photon imaging of in this case, a B78 tumor injected into this 206 reporter mouse, we can actually see nicely this co-localization of Venus positive cells and collagen fibrils. And again, as mentioned in the very nice talk beforehand, these collagen fibrils, can be imaged with second harmonic imaging, and you can see that they have this very linear orientation, kind of on the edges of the tumor mass. As you can see here, this being kind of the interior, and then this being the outer, this rim. You can see the close association of these Venus positive macrophages with these collagen fibrils. And indeed, even towards the interior, you may see there are these stray kind of fibrils of collagen. And again, these these Venus positive cells are, again, especially kind of localized nearby in the neighborhood of these fibrils. So obviously now we have regions of interest. We'd like to understand what is going on in those areas of high collagen buildup and this intense clustering of these Venus positive macrophages. Right. So we took another one of these B78 tumors within this reporter mouse and sectioned that, and these sections are taken 10 days post injection. And what you can see is there's this kind of tumor core, this tumor being M cherry positive. Then you can again see the rim of Venus positive cells. And there's based on the kind of amount of ratio of the amount of 206 Venus signal compared to the actual tumor signal, this M cherry, we're able to find 3 regions. We kind of defined a rim, which or outer section, as we're going to call it. And you can see here, it's a very densely packed region of a lot of this Venus signal. These cells seem to sit almost polarized, oriented in this kind of along the circumference of the tumor. Meanwhile, there's further more Venus cells that seem to infiltrate slightly deeper into the tumor, and they don't follow this kind of circularly oriented fashion. So we define that as the mid, and then areas where they're very sparse Venus towards the interior of the tumor, we define as that intersection. Inner region. So during our ZipSeq approach, we're able to label the immune cells within all CD45 positive cells theoretically within these 3 regions, and from there we're able to extract this information. We found all these different populations of macrophages. I'm sure anyone who follows the field will recognize some of these marker genes. So we have the myeloid arm here, and then of course the lymphoid arm over here. And what we're able to do is overlay the regional assignments on top of this. And you can immediately appreciate that there's significant heterogeneity in where these different populations are localized. If this were a well-mixed system, then when we overlay these signals, you would see, just a whole mix of red, green, and blue. But instead, what you see are clusters that are intensely, let's say, blue. In this case, some of these clusters, areas that are very red, such as these populations here, and areas that are kind of more mixed or more greenish, I guess, in their composition. Each implying that they kind of preferentially localize to one of those regions that we previously defined. Indeed, if you look at the regional compositions, you can appreciate that some of these populations are heavily biased. The colors appear to have been reversed. Anyways, unfortunately, yes, the colors are reversed here, but you can see this population here, this retinal macrophage population here is highly, you'll have to invert the colors in your mind here, are highly localized to the outer region versus, say, these MS4A7 macrophage populations, which are highly localized to the inner compartment or region. So you can see, of course, another thing I like to point out is this naive versus exhausted T cell clusters. You see also this shift from naive to exhausted as you move towards the interior of the tumor here, based on the kind of regional composition. So now we have a bunch of different macrophage populations and their particular gene expression profiles. So we can actually zoom in on some of these populations, such as this MS4A7 macrophage population. Again, this one that's much more inner localized. And upon doing gene ontology analysis of the genes that uniquely define this population versus the other macrophage populations, you get things in this case such as immune response activating cell surface receptor signaling pathway, this complement receptor mediating pathway, and so on and so on. And this first geoterm kind of encompasses like both complement activation and FC receptor binding. So then from this, we can start to get a clue as to potentially what this population macrophage is doing, and try to understand why it's doing that, doing this role towards the interior of the tumor. But then I'd like to actually refocus our attention to these retinal macrophages. So this blue population up here in this corner. Although it's relatively rare compared to the other macrophage populations, one thing we noticed was that it's very, very strongly localized towards the outer compartment, just along the rim of the tumor. And when we do our geoanalysis of its unique identifying genes, we see the cellular response to cytokine stimulus. It's kind of a just general catch-all. But then what we thought was very interesting was this kind of enrichment for these lipoxygenase and lipoxygen biosynthetic processes, which generate these inflammatory molecules. And the reason we call these retinal macrophages is because one of the genes that really defines them is this retinal or resistin-like alpha, also known as RELM-A or FIZ1. It's an interesting gene. It doesn't really have a human ortholog, but there's been quite a bit of literature published on retina's role in potentially talking to fibroblasts and promoting a more fibrotic role for those fibroblasts, increasing their expression of cross-linking enzymes such as lysyl hydroxylase. And then, of course, I'm sure if you remember, the whole original intent of this was to segment based on this 206 reporter, which is driven, in this case, the gene name is MRC1. And so if we overlay that MRC1 expression, you'll see that it actually covers quite a few macrophage populations. Not only does it cover the R-retinal population, but also this APOE macrophage population, a little bit into the proliferative macrophages. So this kind of illustrates the advantage of kind of combining single cell technologies with imaging. Had we just dissolved this tumor without any knowledge of the regional localizations, you would see, of course, all of these are potentially venous positive cells, these MRC1 positive cells. But only with the overlay of the spatial information are we able to say that this MRC1 positive population is specifically localized to the rim versus all these other populations that are not. So I think that helps to illustrate why kind of combining those two data streams is potentially very useful. And another interesting thing about this retinal population is there is this high expression of these signaling molecules CCL6 and 9 amongst other genes. So we're kind of following up on the expression profile here to find ways to basically be able to mark this population by flow cytometry in the future to do better quantification, and then potentially ways to deplete this population as well. Another cool thing you can do now with the spatial information and the single-cell RNA-seq information is we can actually look at the correlation of expression patterns between two different compartments. In this case, we're going to look at this mono-MAC lineage and then the CD8 T cell lineage at the same time. And we can look at their expression profiles for certain genes or signatures of interest based on the region. As we move, in this case, in red, from outer, mid, and inner. And we apply exhaustion signature, we see the same phenomenon we saw in the PYMT model, whereby as you move towards interior, the CD8 T cells show this progressive exhaustion signature. And what we also see is that within the monomacrophages, we see this drop off in this antigen presentation signature as you move towards interior. And this, as we move towards interior, these monomacs acquire this expression of certain markers that the literature has come to use as like kind of your classical TAM markers. Although we would argue that there's really no classical TAM markers. There's so many different populations. But in this case, this complement receptor C1QC. TREM2 is another popular one. However, not all genes show this nice uniformly increasing profile. In this case, TREM2 seems to actually peak in the middle region versus the outer or inner. And then, as I mentioned, one of those marker genes, another one of the marker genes, MS4A7, shows this very progressive increase, very well correlated with that acquisition of exhaustion signature. And then, of course, we want to, going back to kind of the M1 terminology, if we apply that M1 signature here to the macrophages, we see this inverse of that, this very progressive loss of that M1-like signature as we move towards the interior of the tumor. And indeed, another thing we noticed was that within these exhausted CD8 T cells was increased expression of the CSF1, which we theorized could represent a communication axis promoting differentiation and potentially proliferation of these macrophages towards the interior. So the CSF1 expression increases as we move towards interior within CD8 T cells, and we see this progressive loss of this antigen presentation like signature, and this increase in the kind of scoring for this S phase and G2M phase in the macrophages, basically indicating potentially more proliferation. And then I'd like to highlight kind of a really nifty tool we've been using, the CellChat-DB, which was just recently published. It's a very useful tool for studying cell-cell interactions in single-cell data sets. But then one thing we thought about was that there's this, it infers these cell-cell communication pathways based on gene expression, but we felt that we could give better confidence to the communication axes that predicts with the overlay of spatial information. Because, for example, cell-cell like juxtacrine signaling, you expect if you predict this interaction between cell and interior and the exterior of the tumor, that's probably not as likely as interaction that's happening within the same region. Of course, with certain secreted factors, you can definitely get more interaction between different regions. But generally speaking, we expect cells within the same region, interaction within the same region to be potentially more likely than those from spanning the whole tumor space. So what we can actually do is you can look at the kind of incoming and outgoing interaction strength of these all these signaling pathways, and then look at that within the outer region and then within the inner region, and you immediately appreciate that there's some populations that dramatically shift in kind of the amount of signal they're receiving and sending. What you can see is, like for example, that retinal macrophage population, because it's I mean, it's not really seen in the inner population. Of course, there's very little signaling to acquire there. These APOE macrophages, meanwhile, seem to be receiving and sending signals quite well in both outer and inner regions. And of course, each of these populations starts to shift a bit. And then what you can do is you can look at the amount of information transfer for a given pathway. This information transfer is kind of like a sum total of all the weights of these kind of arrows, these bands that connect different populations. And then you can look at ones that are found basically that are rich towards the outer region, and then within the inner region are another set of the pathways that kind of have more information transfer. So for example, in the exterior, you see CX3C, you see MHC2, fibronectin, and towards the exterior, towards the interior, you see things such as IL-2, and there's, I believe, CSF, and there's TNF. So then what you can do is let's see who's talking to whom. So let's pull out the fibronectin network and see for the outer versus inner. You can see these inflammatory monocytes are one of the senders basically for this pathway. Basically implying they lay down fibronectin, which is received by integrin receptors on other cell types. But then in the outer area of the tumor, you can see this retinal macrophage population pops up. They're major producers of fibronectin, and as predicted here, they basically communicate with all these other cell types. And then we can going towards the interior, let's pull out TNF and take a look at that. You can see is that there's a major sender of this is APOE macrophages. But then as we move towards the interior, there's many more MS4A7 proliferative macrophages, increasing the weight of these interactions, these sending interactions. So we see that they're the main producers and the senders of this TNF signal. And then, for example, CSF, as I hinted before, it's produced by these exhausted CD8 T cells. And indeed, if we move towards the interior, you see this much more weight of signaling stemming from these exhausted CD8 T cells to these macrophage populations. So I think this is a nice demonstration of what additional information you get from spatial information and take tools that have been developed for cell-cell communication, kind of marry the two. And indeed, that going back to that retina positive macrophage population, we wanted to verify this is indeed true. So by IF, you can actually see that in this case, retina, also known as realm A, stains nicely and is pretty much restricted very nicely to the exterior of the tumor. And if you look closely, it is found within CD11B positive cells. So it's not just being expressed on like some other stromal cell type. And indeed, if you look at CD11B positive macrophages just a bit into the interior of the tumor, they seem to actually lack this retinal expression. So what we have here is a population of macrophages that has a very distinctive spatial profile and a very distinctive transcriptional profile. And so kind of narrowing down its role is of immediate interest to us. And then kind of translating this to human context, we were able to, I'll briefly introduce this idea, which is this immunoprofiler initiative. The idea was to basically characterize a bunch of human tumor biopsies. And here they've actually sorted, this immunoprofiler initiative here, UCSF, they sorted these different compartments of cells and performed bulk RNA-Seq. And using this, they're able to generate a bunch of features which they basically used to categorize these tumor biopsies into 12 different what they call archetypes. So these archetypes kind of define tumors that share a certain profile of gene expression. So you can see there's different like immune-rich CD8 and immune desert, myeloid-centric, T-cell-centric, and so on and so on. I'm going to move through this relatively quickly. If you are interested in the further details, this paper is on bioRxiv. You just need to search like PanCan and the first author, Alexi Combs. But we wanted to mine this data set using expression signatures.
we should probably wrap it up soon so we can allow for some questions.
Oh, yes.
We have to finish at 1.
Oh, yes. This is the last slide. And so we wanted to mine the data set. And so we took the top unique differential expressed genes for that retinal population. And what we found was that, so that's this set of genes right here. You can see that they're enriched for the myeloid-centric DC2 and immune desert CD8 bias, these two archetypes. And if we also look at in the tumor compartment for these archetypes, we see that, so if we look at those two archetypes, what we see is that they're, one thing that they share, one of these modules that they share is this fibrosis module. And of course, there's some other modules that they also seem to share as well, such as the cell cycle ones. But we're very interested in this fibrosis module and potentially understanding if this represents a population conserved between mouse and human. And our current hypothesis is that they're instructive. Not only do they lay down fibronectin, but they may be instructive for other cell types, such as fibroblasts in ECM remodeling. So I'm just going to skip through this then. It's kind of a side module. So very quickly, a plug. If you want to adopt SIP-seq approach, we put more details on the exact workings of the approach and all the parts, what you need. It's on our website, and you can see there's exemplary data and protocols and the more detailed parts list, what can be found on the Nature Methods paper. And with that, I'd like to thank the lab, especially Arjo, who developed that reporter mouse, and the AminoX co-labs, and the Reeves lab, which unfortunately didn't get time to talk about, the mosaic tumors, and our funding sources. And with that, I'll take questions.
Amazing, thank you, Kenneth. So we have a question from Chris Madsen. He said, did you find a correlation between macrophage subpopulations and linearization of collagen?
Yeah, that's definitely a question of interest. So as you saw there, it does seem like those venous positive macrophages are closely associated with those linear stretches of collagen, but we still haven't reached the level where we can say there's a causation. So that will require some potentially genetic ablation experiments, which with the DTR built in, we think is definitely possible. So one idea is to deplete those macrophages at different time points during tumor progression, and then and thus image the collagen fibrils. And of course, now I realize, now after seeing your talk, I see that if we can actually get very, very good details on the structure that ECM using your approach. So definitely be interested to try your approach on our tumor models, following different following depletions at different time points, potentially the tumor growth.
And the question from Chen Zhao, how many cells can ZipSec capture from each sample?
Well, that definitely depends on the kind of richness of immune cells within a section. So, I mean, when we perform this on lymph nodes, you get way more cells than you'll ever need to. I mean, it's chock full of immune cells. Now, tumors are kind of iffy. It depends on kind of the immune infiltration of the tumor. As you saw in that data set, we were only able to acquire about 3K cells in one section. And we're kind of time limited right now based on kind of our imaging setup to doing like one section at a time. However, we're in the process of developing a new microscope right now, kind of the spatial dissection of multiple sections at a time, and thus kind of thus linearly scaling up the cells you can recover. So I'd say for it's hard to say generalized, but in our tumor section, it was about I don't know, like 3-4 millimeters about in diameter, so about a circular section. So we're able to get about like sort out about 5,000 cells, and then with 10x's recovery rate, it's about 2-3K cells.
I'd like to ask Bethan, are your mice so young that they don't have much fat, or how do you image through fat?
The fat is a massive problem for the mammary gland. So it has the benefit of being,
for us, definitely.
It has the benefit of being very superficial as a tissue, so you don't have to obviously go through the peritoneum to do the surgery, and that makes it much easier. But the fat being also like reflective, and the appearing is deeply encased in this light reflecting fat pad, and also the fat casts shadows and rings over the images. So most of my studies are in quite young mice, and obviously they're much easier because than older mice that tend to have fattier mammary glands. It was, even in young mice, it can be challenging if the structures are too deep, but near surface ones you can get really nice images from.
I see. So you basically implant the window, and if you're lucky that the surface is
a little bit. I think most of us who do mammary gland imaging of the normal epithelium or really early, when in the tumors, that's not a problem because there's no fat basically.
Right. The fat gets pushed away in the fully formed tumor, but the developing gland, I can imagine that being a nightmare.
It's challenging and requires a little bit of good fortune sometimes, but it's feasible.
I wish we could clear living mice.
Yeah, there are some groups who have published some things about clearing, mainly in
there was like maybe 10 years ago, even like there was a transparent mouse, but I don't know if you ever tried using that.
No, I'm quite interested in people who have done crystalline knockout mice, so modulating crystalline proteins, which makes things transparent in other organisms, and I think people are really interested in that. Whether it will happen in my lifetime, I don't know.
That would be actually a question for Chris. Did you try to decellularize the whole mouse?
No, not the entire mouse, but the majority of parts of mice, right? So, and the reason is that you need a good pressure in a specific organ. So if you have too much high pressure, you ruin the organ, right? So if you need to get the soap out in the most, distal part of the animal, you need a very high pressure, and you will ruin the organ nearby. So you need to have it balanced. So that's why you clamp the vasculature, so you concentrate decellularization of certain part of the animal. So for example, if you want to go to the brain, you will operate up here, and you will perfuse up in the brain only, and not the rest of the body, you see.
So that's also good. I mean, how much antibody do you use, or you can recycle it?
No, the antibody, you can do that afterwards. So you can just actually take the organ out, and then you can soak it in a.
But still, you use a lot of antibodies, right? You need tons of it.
Yeah, we'll use a lot. So of course, it depends on what the purpose is, because if you have a big lung organ, maybe you don't want to image the entire lung. You can take a lobe, and so forth, and you can reduce a bit the cost if you want to. But
related to that, Chris, in the mammary gland, which I consider not highly perfused compared to some other tissues, like did you struggle to decellularize the mammary gland, or was it relatively okay?
No. I mean, when we start struggling, which is the big problem, is that when you have a very dense tumors, where you don't have a good vasculature, right? So you need the vasculature. This is a process where you use the native vasculature to perfuse the organ. And what happens sometimes in some tumors is that there's poor vessels, and there you need longer time to do it. So it's not something which takes a lot of time.
Can I ask you about the? Oh, sorry. Go ahead.
Sorry, I have a non-tumor question. So did you do this in the involuting gland by any chance? Because you showed some beautiful images of the lactating. I'm just wondering about the architecture of the involution gland, because it's much harder to image that normally.
It was a proof of concept, actually, that part.
I have a question for you about the gut. You didn't show the decellularized gut in your papers, and we tried it, so no problem to decellularize. But as was Bojana mentioning, it's all fluffy, very hard to handle or to do anything without after decellularization. So did you guys try it?
Yes, I think Alejandro played around with it. And as you say, it might be very fluffy. Some organs are very fluffy. I mean, the brain is nearly impossible, right? Because you just, it's just, liquid pretty much, right? And so this is issued sometimes for some organs. But generally speaking, so you don't maintain a good structure of some organs. But when you place it and you look at the ultra structures resolution, I mean, on single fibers, they seem to
right. Then yeah, exactly. Yeah, this I agree. But keeping the structure, let's say the villi, it was impossible.
What was very interesting is that we tried to image some organs 6 months later, and they and we did the staining, we kept it for 6 months, and we did the staining and we did the imaging 6 months later without restaining it, and it was surprisingly well conserved, and we didn't even have to restain. So we could actually stain and image the same tissue 6 months in between. Very good accuracy. So if you maintain no bacteria infection and stuff like that,
And I also have it was interesting observation that you show 2 vessels next to each other, one labeled with collagen 4, one with laminin, I think. Yeah. That was striking that they were not colocalizing. But did you try? It was laminin 1, if I remember well. Did you try staining with other lamins? It's just that the different isoforms, or you think they actually have less laminin?
This is a good question, because of course you base your imaging on an antibody finding an epitope, right? So the question is, do that epitope, is that hidden somewhere where you can't get access? And we can't really know that, right? So if you have to do anything, you'll have to do some ratiometric proteomics to see if there's really differences in, but then you have to pick up the vessels like one by one. But we tried various different laminins, gamma 1, gamma 2, and different things, and we do see surprising differences. But it's not something we really systematically gone into. But of course, it's surprising when you see that exactly that image. And I think old-fashioned ECM people from the 80s and 90s would probably don't like that concept. You have the idea that it's all the same, like the basement membrane is the same, but it's not. It's so clear, right? So, so this is probably something one should really go into detail and figure out.
Yeah, I agree.
I mean, Janine had we had a collaborative paper where they looked at netrins in the basement membrane.
Yeah.
And that's just one molecule which is not considered to be a classic basement membrane, but when that's not in the basement membrane, it changes the stiffness of the basement membrane, right? So the size, the stiffness, the holes in the basement membrane is changing just by having a protein called netrin 4, right? So there's clear differences in basement membrane structures and organization in probably even different organs, right?
More questions from we lost a lot of participants already, so it's time. Maybe question for Guillaume, where are you going with your chamber? In which step you are now? Can people actually use it? How, if they want to collaborate with you, how does it work? I mean, I collaborate with you, so it's easy, but others.
Just contact me, Bessan, or Sylvia, and then we send window. Finally, we can produce a lot. So a lot. It's easy to produce. So
you're pretty fast now, Leo.
Yeah, 5 minutes per window approximately now. So
can you also make copies of your artwork? Is it yours behind you? You have a mouse painted.
It's my father's work. That it's a counterfact, I think.
It's a what?
Counterfact.
Very nice.
Okay.
So sorry, I would like to apologize to Kenneth. I was lost for a bit of your talk, so I really didn't follow on the end. I was coming in and out. Sorry.
But Guillaume, can I ask you a question? How long time did you maintain these window chambers in the animal? For how many weeks?
We went up to 35 days, and because the PDMS is certified for 30 days, I didn't want to push more to avoid like degradation of the silicone or things like that. But seriously, at this point, I didn't precise that during the talk, but there is some holes around the frame. So there is fibrosis of the window within the tissue, and that's very tough to I didn't show this video because it's disgusting, but you can pull the mouse by the window. It's totally fused with the skin.
Okay. So you have a lot of fibrosis and angiogenesis around the rim.
Yeah.
Mm.
So is the medical grade PDMS much different than just PDMS we use for our microfluidic devices? Is it just more expensive?
It's not more expensive. Finally, it's cleaner, and because I had some discussion with a PDMS specialist, and it's just that they ensure that 100% of the catalyzer, which is often platinum or stuff like that, are catalyzed. It just avoids that there is a release of metals like platinum in the body.
I see.
Overall, I think it's exactly the same. It's just a matter of testing and you know, the ISO stuff, just to say cost twice the price.
Okay.
There is also one question for Chris from Miranda Soder. Did you image the ECM within the long bones like tibia or femur?
No.
Yes, is no.
No. Good idea.
Okay.
Okay, it seems like we're all done with the questions from the audience and I guess from the panelists as well. In which case, thank you all for joining us today. And next session coming June 11. And we're going back to the Europe East Coast time. So it's going to be starting 2 hours earlier than the one today.
Thank you all.
Thank you very much.
Bye.
Bye.
Have a good weekend. Bye-bye.
Thank you.